Hello, good evening, everyone. Welcome back to day three of our 1 week hands on workshop on network pharmacology, molecular docking, pharmacokinetics, and molecular simulations. On behalf of Time Phytobiomet Private Limited and AdGrow Foundation, I warmly welcome all our speakers and participants. Thank you for joining us once again. We hope you found yesterday's session on molecular docking and software installation useful and that everyone has successfully installed the required software. So, today we will continue our workshop with two very important topics. One is pharmacokinetics and other one is molecular simulation. In today's session, you will learn the basic concept of this technique and understand how they are used in molecular drug discovery and research. This session will prepare you for the practical session in the coming days. I encourage everyone to actively participate and ask questions during the discussion session. Now, without taking much of your valuable time, it is my pleasure to invite our respected speaker, Dr. Ghansham, to deliver today's session. Sir, we are delighted to have you with us once again. Over to you, sir. Hello, good evening, everyone. So, I hope you all are doing well. Welcome back to day three of our 1 week hands on workshop on network pharmacology, molecular docking, and pharmacokinetic and molecular simulation. On behalf of Time Phytobiomed and AdGrow Foundation, I warmly welcome all our speakers and participants. Thank you for joining us once again. We hope you found yesterday's session on molecular docking and software installation useful and that everyone has successfully installed the required software. So, today we will continue our workshop with two very important topics, pharmacokinetics and molecular simulation. In today's session, you will learn the basic concept of these techniques and understand how they are used in modern drug discovery as well as in research. This session will prepare you for the practical session in the coming days. I encourage everyone to actively participate and ask questions during the discussion session. Now, without taking much of your valuable time, it's my pleasure to invite our respected speaker, Dr. Ghanshyam Parmar, to deliver today's session. Sir, we are delighted to have you with us once again. Over to you, sir.
No, sir. No.
I think now I am audible.
Yes, sir.
Okay. Sorry for the interruption. So actually, there is a one minor correction in the today's schedule. As I'm sorry for the little bit more modification that this section is divided in a three part actually. First part I will cover with the pharmacokinetic property. Second part will be covered by the J, and in third session or in third section we will help you to if there any troubleshooting in your installation of your software, whatever the we software or discussed in yesterday. If you find any difficulty for installation or if you have any troubleshooting, so in last part of the session we will help you out to all related all troubleshooting. So this is just a minor modification. So I hope you will or you will enjoy this today's session. So let's start by today's session. I hope my screen is visible to all. Okay, so let me introduce a topic that is a pharmacokinetic. So basically, pharmacokinetic is a actually a phenomena or a process where the what happened when we administered any drug to our body, either any of the route like oral route, yeah, any uh, intravenous, intramuscular, subcutaneous, inhalation, transdermal, any of the route we have administered any drug, then what happened to that drug? Okay, so there are various stages of a metabolism of this drug or processing of this drug in our body. So this entire phenomena is called as pharmacokinetic, which include absorption, distribution, metabolism, excretion, and lastly, if there any toxicity. So basically, pharmacokinetic describe the time course of drug in the body and how the body absorb, distribute, metabolize, excrete, or tolerate, or I can say the toxic effect of the drug. Okay. Why this pharmacokinetic parameter is uh, necessary to understand in a drug discovery? The question may arise in your mind, because ultimately, whenever we are designing any new drug, it must be having the all these good properties like good absorption, good distribution, having a fair metabolism, having a proper excretion, and no side effects or no toxicity. So by this compiling this property, a finally a druggable substance can be generated. Okay, so to qualify of any ligand or a molecule as a drug or druggable substance, these all properties should be understood. Okay, so absorption means how the drug is entered into the bloodstream. Okay, that depend upon the how we administer the drug or how what route we choose for the or what kind of formulation we are using. Okay, what is the physicochemical property of a drug molecules like solubility, permeability, and whether it is stable or unstable molecule. Based on that, absorption is dependent. Most commonly route that we use for the administration of any drug primarily is oral route. Majority of drugs should be qualified for the first oral route. Then second is intravenous, intramuscular, subcutaneous, inhalation, transdermal. These all kinds of administration route is available for the drug. The route of administration mainly decide based on this all the properties of the drug that already I described, like solubility, permeability, stability. Most of we all are aware about the various class of the drug, like BCS class 1, class 2, class 3, and class 4 drug, which totally depend upon the solubility and permeability of a drug. Some drugs are highly soluble and highly permeable, while some drugs are highly soluble, low permeable, while some drugs are class 3 like low soluble, high permeable, while many are class 4 drugs which are low soluble and low permeable. So these all kind of property we need to understand while we are administrating or we are uh, thinking about the drug discovery. Distribution is how the drug is distributed into the body, how the drug is spread into the body. So reversible transfer of a drug from blood to tissue and fluids, which through the blood flow. Protein binding, lipophilicity, action of transporters that decide the how drug is distributed into the body, like plasma protein binding, volume of distribution, blood-brain barrier, tissue affinity. All these factors affect how the drug is distributed into the body. Once the drug is properly distributed to the body and reaches to the binding site and available for the action, that is called as bioavailability. Once the drug is bioavailable to the body, it gives action. After this distribution and bioavailability, the drugs need to be metabolized. Okay, so how drug is chemically changed. Sometimes the metabolites are also active. Say for example, prodrugs. Most of the prodrugs are active once they are metabolized into the body. Initially they are inactive, but once it goes to the bloodstream, it get metabolized into its active form. So this is the beauty of this kind of drugs. Most of the metabolism occur into the liver, where the liver enzyme act and by various processes like oxidation, reduction, conjugation, hydrolysis. All these phenomena are responsible for the metabolism of a drug, and drugs are metabolized into its metabolites. These metabolites sometimes may be active, inactive, or maybe some metabolites may produce a toxic effect also. Accumulation of this metabolite may produce a toxicity. By combination of drugs, sometimes it produces the drug-drug interactions. As we know that in our system, we use sometimes polypharmacy, multiple drugs simultaneously. So we need to check whether the drugs are interacted with each other and produce a toxic effect, whether the metabolite are producing a toxic effect. Next step is excretion, where the drug is and its metabolite are leaving from the body, thrown out from the body through various routes. Like major route is urine, other route is biliary or fecus material, lungs, sweat, saliva, milk. All these routes are responsible for the excretion of the drug. So we need to think that, or we need to keep in mind that how the drug is being excreted or how metabolite can be excreted. So for that, we need to keep in mind the various factors like renal function, biliary excretion, and the drug properties also. Then we need to think about the toxicities of or tolerance of the drug, how the body responds when the unwanted and harmful effect due to a drug or its metabolite, it's called toxicity, which depend upon the dose, duration, and individual variability. If drug is given in a higher dose, then the rate of excretion affected. Then the drug may be accumulated into the body, or metabolite may accumulate in the body and produce a toxic effect. Lesser the dose, maybe it does not produce a sufficient effect also. So all these aspects we need to consider, or we need to check, or making the our ligand a druggable substance. So all these parameter we need to understand first, and then we need to check it. So key pharmacokinetic parameters or key terminology mainly used are Cmax, which means a maximum blood plasma concentration. If you look into this curve, where here the concentration of concentration of drug in a blood or in a plasma is considered as maximum concentration, is a Cmax. So time to reach to this concentration is called as a Tmax. The time, whether it take 45 minutes, 10 minutes, or in a second sometime. Mostly conventional tablets, what we are taken in a oral form, like paracetamol tablet, metacin, which take 30 to 45 minutes at its Cmax concentration. While IV drug are immediately they reaches to the Cmax concentration, because it bypass all the metabolism, all the first pass metabolism and everything. So it reaches to the Cmax immediately. Then area under curve, this area where the plasma concentration is distributed throughout the body, that's called as a area under curve. It is called as a total drug exposure. Then one more term which is most familiar, that is T half, which is called as a half-life of a drug, which means that a time to drug or time to reach a 50% of concentration of drug is removed is called as a T half, or half-life of a drug. Then CL means clearance volume, volume of plasma required, or volume of plasma that clear per unit time. Then volume of distribution, VD, how the drug is distributed into the body, that is called as volume of distribution. So any ligand or any druggable substance, heat or lead, should have ideal ADMET profile. Then it can be considered as a druggable substance. Good oral absorption, because we are most giving priority to the oral route. Then we If oral route is not feasible, then we go for this intravenous, intramuscular, and subcutaneous route. Appropriate distribution to the target tissue, stable metabolite, no with a lesser side effect or no side effect, safely and efficiently excretion. Sometimes many of drug can during excretion produce toxic effect to the filtration system or harmful to the kidney. Some of the drug are excreted into the milk. So while we designing any drug for the lactating mother, we should have to consider this excretion where sometimes the drug is leached out into the milk and it will through the milk get to the body of the infant. So there may produce a side effect to the infant. So these kind of factor we should consider. Minimal toxicity and good balance and effective and safe drug. So these all pharmacokinetic properties decide the how your ligand, how your hits or leads make druggable substance. What are these other factors which affect the ADMET properties, like its physiochemical property like molecular weight, log p value, pKa? All these parameters or all these properties affect these all ADMET properties. Dose, route, age, gender, whether the genetic material or genetic footprint, disease state, and drug-drug and food-drug interactions. These all are factors also affect the ADMET properties of any drug. Another concept we need to understand, that drug likeliness concept. So what is a drug likeliness? It refers to the structural and physicochemical property that influence the ability of a compound to become an effective oral drug or effective drug. So as we discussed the various physicochemical properties that matters for the ADMET properties. So these physicochemical properties are responsible for the drug likeliness. So while we designing any molecule or synthesizing any molecules, or we are finding from the natural sources, whether it is a synthetic compound, natural compound, biological product, biosimilar, whatever it is, we need to check for the drug likeliness. Why we should check? It improves the probability of a clinical success. It reduces the last stage failure, because most of the drug are failed in a pharmacokinetic. Then it helps us to guide a lead optimization. Suppose we are synthesizing or designing a molecule which having the property, a toxic property. So based on this physicochemical parameter, we can alter or we can replace certain functional group and we can redesign our molecule. That is called as a lead optimization. By checking this property, we can alter the structure and it gives you a drug-like substance, a good lead or good hit. And by checking this physicochemical property, it ensures us a better absorption, distribution, metabolism, excretion, and safety also. So what are these physicochemical properties or parameters? Before that, we need to understand a rule of 5, or that is called as a Lipinski rule of 5, which is an empirical rule to evaluate for oral bioavailability of any compound. What is bioavailability? Bioavailability means, anyone know what is the meaning of bioavailability? I already explained you. Yes, please. Please answer the question. What is the meaning of bioavailability? I already explained you. Just
Amount of drug which is available in the blood or plasma.
yes, exactly. Thank you for your answer. So this depend upon the Lipinski rule of five, first and foremost. So what is Lipinski rule of five? So five means molecular weight less than 5 Dalton. 500, sorry, 500 Dalton. Log p value less than 5 means lipophilicity. Lesser the lipophilic drug, more will be the absorption. Hydrogen bond donor must be less than 5. And hydrogen bond acceptor less than 10. This is called as a Lipinski rule of 5. At present, in many of the company or many of the research institute modify this rule from rule 5 to rule 3. Molecular weight less than 300, lipophilicity less than 3, and hydrogen bond donor less than 3. So many of company are or many of the institute are now using this rule. So during checking of this property, if you find any violence in this rule or more than one rule of violence, it indicates it have the your lead or your compound might have a poor absorption or poor permeation. Okay. So I have cited one example. You can check it. Molecular weight less than 500, log p value less than 5, hydrogen bond donor less than 5, and hydrogen bond acceptor less than 10. So here zero violence of rule. So this compound can be having a very good bioavailability, oral bioavailability, and having a good absorption and permeation also. So the question may arise that how I can find my desired molecule having this kind of violation, or maybe it passing all the rules and regulation of or rules of Lipinski rule of 5. How do I check? Question may arise in your mind. So let me give you a brief demo of how to find this kind of property. Just let me share my entire screen so that you can visualize all tabs which I am using. So for that, you need to go to the Swiss ADME portal. Yesterday, someone asked me that how to draw a structure. Okay. So here a panel is been given. It is associated with the Chemex zone. So you can draw structure from here only. Okay. Or other than this, you can go to or you need a various software, like one is called as a Candro. you can draw a structure from this also by using this kind of software, CamDraw, CamExon, then CamSketch. Suppose I want to check the ADMET property of this molecule, which I have designed, say for example. Then I have selected the structure, go to the structure and convert, or sorry, go to the edit and copy as smile. Again, I am repeating it. I'm drawing the structure. I'm selecting it by clicking on this arrow. Then in edit, copy as smile. So smile is a nothing but is a textual representation of any of the chemical structure, because software doesn't understand a 2D or 3D interpretation or 2D or 3D representation of any molecule. So we need to give a simplest way of representation of our structure. So that is called as a smile. There are different kinds of smile, like canonical smile, then incal smile. Okay, so that we need to copy. So here I am taking a canonical smile, which I am copying it. Then go to the Swiss ADME and pasting the smile. Then Control V. Once you have pasted the smile, then you have to go to the run. Once you're done, it will finish fraction of second and it will give you a all property of your physicochemical property of your drug, of your molecule. Like starting from molecular weight, number of heavy atoms, then rotatable bond, hydrogen bond acceptor, donor, total polar surface area, lipophilicity, here log P value, this one, octane water. O means octane and W means water. So octane water log P value should be considered. Then you need to check the water solubility. Then here pharmacokinetic parameter is also be given, like GI absorption, BBB permeation, mean how the drug can be cross the BBB, blood-brain barrier. Then PGP flux, then metabolism, like CYP. And at lastly, you will find that here you will find the Lipinski rule violations. So here one violation is there. And it at last it gives you a lead likeliness. Okay. So all these parameter you can download also by just clicking on this icon called as CSV. So your CSV file will be, CSV means comma separated file, which is a, you can open with the Excel. And here in Excel, all the whatever the parameter it has been checked from through this portal or a database, it imported into this Excel sheet. like SMILE, molecular formula, molecular weight. Types of heavy atoms, rotatable bond, hydrogen bond acceptor, donor, total surface area, polar surface area, log p value. Everything is imported into the Excel sheet. So here in this section, not only you can take a single, whatever the number of smiley you have. Say for example, from the different database like zinc. Okay, from zinc database, if you are downloading the entire database, other like NPACT, that is a natural product anti-cancer database, then coconut, which is also a database of a natural product, then our own Indian database is also available, that is IMPPAT. Indian Medicinal Plant and Phytochemistry and Therapeutics. There are many of databases are available. Campbell, okay, for synthetic compound, Campbell, sorry, database is available. PubChem itself is again a database. Campbell, then PubChem. Say for example, I am writing here routine. Just click on the structure, or sorry, name, and you will find all the property. So here, this side, you will find the relevant, whatever the relevance resources you require, you can directly check from these also. There are different kinds of smileys has been represented. So you have to copy this smiley and paste into the this portal and then run. You will find all physicochemical property of routine, like molecular weight, heavy atoms, rotatable bond 6, hydrogen bond acceptor, donor. You might be think that the routine is totally violating the Lipinski rule. Okay, 3 violations is there. Molecular weight is more than 500. Number of donor is 10. Okay, but still it is a use as a drug. So there is a one catch here in this Lipinski rule of 5. It's sometimes not applicable to the natural drug or natural products. Say for example, if I just talk about the podophylotoxin, let me just share you give you example. A routine itself is a very good example where it violating the Lipinski rule of 5, but still it is considered as a druggable substance. Okay, so this might be clear to you that how you can derive all this data, which can be helpful for your ligand optimization or make the drug or make your ligand drug-like substance. So there are some important rules also that we need to understand. That rules are Weber's rule, Egan's rules, Gauss filter, and Maggie's rule. Weber's rules gives an idea about the if the total polar surface area means how your drug is containing a polar groups. So total surface creating due to this polar functional group that should be less than 140 angstrom, and rotatable bond must be less than 10. According to this Weber's rule, if any ligand follow this rule, it might have a good oral bioavailability. Similarly, other rule like Egan's rule, where the log p value less than 5.88 or less than 5 and total polar surface area less than 131 angstrom, having the good absorption. Gauss filter, where it described that molecular weight between 160 to 480, log p value minus 0.4 to 5.6. Hydrogen bond acceptor between 20 to 70 and hydrogen donor between 0 to 5, having a drug likeliness for a small molecules. So all these rules we should have to consider according to our requirement and according to our aim and objective. So in general, ideal property range is, as I told you, that molecular weight between 150 to 500 Dalton, log p value minus 0.5 to 5, total polar surface area, okay, that is of in between of 20 to 130 angstrom, hydrogen bond donor between 0 to 5, hydrogen bond acceptor between 2 to 10, and rotatable bond in between of 0 to 10. So all this ideal property may decide the along with the solubility and permeability. So balancing the in solubility and permeability make the ideal drug-like substance. As I told you earlier that based on the solubility and permeability, all the drug are classified in a 4 class, which is called as BCS class or BCS classification. Okay, you might be aware about the BCS classifications. Which is called as a biopharmaceutical classification system. Okay, which is four class of compound or four class of drug are there. Class 1, class 2, class 3, and class 4. Where class 1 are highly soluble, highly permeable. Class 2, low soluble but high permeable. Class 3, high solubility, low permeability. While class 4, both the solubility and permeability is low. So according to this classification and this ideal property make the drug or make the ligand as druggable substance. Along with this, you can assess all this property not only on Swiss ADME like PKSIMS, Mol Inspiron, ADMET Lab, QuickProp, Drug Discovery Studio, all this other portal you can check it for these kind of properties. Okay. For the toxicity concern, we need to go to the Protox. Protox portal. Protox 3.0. This portal give you idea about the how to predict the toxicity of any of the molecule. So go to the tab toxicity prediction, tox prediction. Then paste your canonical smile. So already I have copied the smile of routine. So I paste it, then automatically it will draw the structure by just clicking the on a smile. So smile will convert into the structure. Okay. So this is the structure of routine. Then you have to select what toxicity you are looking for. You are looking for organ toxicity. You are looking for toxicity endpoints, or there are other toxicity and metabolism also be described here. So according to your aim and objective, okay, or according to your behavior or property of your compound, you can select the toxicity parameters. So I am currently now just checking the hepatotoxicity, neurotoxicity, nephrotoxicity, respiratory, cardiotoxicity. Then carcinogenicity, immunotoxicity, mutagenicity, cytotoxicity, blood-brain barrier crossing, then clinical toxicity and nutritional toxicity. I'm just checking this all, which is sufficient for me to filter out the any of the molecule. Then check or click on the start tox prediction. The new window will generate and it will gives you again a pharmacokinetic property here. So you can get the pharmacokinetic property from this tab also, like molecular weight, number of hydrogen bond acceptor, donor, everything you will get here. TPSA, topological polar surface area. Okay, this is the log P value, octane water partition coefficient, log P. Okay, so here also you can get the pharmacokinetic parameter. Also you can get the LD50 of this compound. LD50 means lethal dose. Based on that, you can decide your therapeutic dose. Okay. 1/10 of a or 1/5 of the LD50 would be ideal for the your therapeutic dose. If you don't know about a newly synthesized molecule or LD50 of newly synthesized molecule, then you can use this portal. It is a estimated or predicted LD50. It is not an actual. For actual, you have to perform acute toxicity study in an animal. The 50% of the animal, if died at whatever the dose, that dose is considered as LD50. You have to start from smaller dose to the larger dose at a different interval. It also gives you a toxicity class. Class 5 is a safe one. Class 6 is also safest, but class 1 toxicity is considered as a danger one. Once you go to the bottom of your web page, you will find your toxicity profile, like organ toxicity, particularly whatever we have checked, that is hepatotoxicity, nephrotoxicity, neurotoxicity, nephrotoxicity, all the toxicity, whatever we have checked before entering the smile, that all are observed here. So green section gives you a pass the sample. it doesn't have any toxicity as a on liver. It doesn't have any toxicity on neurons, but it having the toxicity in a kidney. Also, there is a chances of a toxicity in a respiratory system. So all similar toxicity prediction you can get. Now just and whatever the previous your pharmacokinetic parameter is there, just copy paste there. And sorry, not like this. You have to copy this your target and transpond here, not paste directly, transpond it to the vertical to the horizontal. Similarly, copy the whether it is active toxicity prediction or inactive toxicity. Just copy it and paste it in a transpons. Likewise, you can prepare entire sheet of your all molecules and numbers of molecule. Okay, hundreds, thousand, millions of molecule you can copy and paste here. And once your sheet is ready, apply the filters. Apply the filter. According to your understanding of all the parameter I have explained here in pharmacokinetic drug likeliness concept. Okay. Apply this filter into this Excel. Like I want to filter the rotatable bond from here. I can filter it from here. Hydrogen bond donor, acceptor. I can apply the rule also. Okay. Number filter equal to, greater than, less than, in between. I can apply the rule accordingly as per my need. Okay. Similarly, at the end, I can apply the toxicity filter here. Suppose I want to filter out whether the my drug should not be cross the blood BBB. Then accordingly, I have to apply the filter. Then it will filter out the molecules. So this is just an overview of how you can filter out your ligand molecule according to the ADMET properties. So better the drug likeliness, better the drug candidate. This is the final outcome of this slide. These are the certain parameters that we need to understand. Already I have explained it, but these kind of parameters are for responsible for the final formulation of your drug candidate. This is just for your information. If you go through this slide, you can easily understand, or I already explained it, so I'm not going much in detail. Okay, so up to this, if you have any query, you can ask me. Then I'll hand over the session to the Jai for explanation of a molecular simulation. If you have any query, you can ask me related to the pharmacokinetic and ADMET properties. Otherwise, we will move ahead for the molecular simulation. Okay, there are few questions. Sir, what if the compound is violating log p value? Okay. Again, log p value if violating, okay, that decide its distribution into the body. So it is our prime parameter that should not be violated. Someone written Dr. Dukes and knapsack. Yes, that might be used for the property. Sir, if we studied neuronal disease, but our compound is showing neurotoxicity, so can we select this compound for further study? No, definitely not. If it is toxic to neuron, then why should I have to study for the neuronal disease? So does BBB permeability be active or inactive? It must be inactive. Because the drug molecule cross to the BBB, then it may okay. Sorry, it may disconnect. Am I audible to you?
Yes, sir.
Okay. Okay. Because there is a someone asked that what if our compound cross the BBB should be rejected? See, that is depend upon your aim and objective. If you are targeting the brain, then your drug should be cross the BBB. If you are looking for the drug should not be cross or should not be reached to the brain, then it should be inactive. It should be rejected. The simplest answer, I think. Sir, how can we prepare ligand library of Lexop compound? Okay. So by using various software, say for example, in some commercial software like Schrodinger, there is a shoot where you can leak prep shoot that you can use for the preparation of your entire library, fragment based library. Okay. Then one of them software called MolSoft that also give you a ability to generate a library of molecules. The other way is that you can download a entire library from these kind of database like PubChem, then Zinc database, where you can download entire library in either SDF file, CSV file, whatever you are required. I think Pratibha, I think I'm answering you in a very good sense. So similarly, if you just look into this here, here in Swiss ADME, oh sorry, I'm not shared my screen. So right now in Swiss ADME, the CYP inhibitor or inhibition or pharmacokinetic property of CYP related is temporarily unavailable. Okay. So for that, you need to go to the ProText. Just from the starting. Then here, metabolism related to CYP. You can check on it and check your prediction. So here you will get the idea about the cytochrome metabolism. What are the reasons that molecule is druggable but it is violating the Lipinski rule? How check the molecule is druggable if it failed the Lipinski rule? This is mostly happen with the natural compound, because natural compound doesn't follow the Lipinski rule. But in case with a synthetic drug, this scenario would not be possible sometime. Sorry, measure of time. Another answer of your question, that how to check the molecule is druggable if fail Lipinski rule, then we have to go with the in vitro and in vivo testing. That, or rather I can say the acute toxicity study, that will finally decide whether the drug is uh, toxic or not, or druggable or not. Ultimately, experimental validation is necessary. These all are prediction. It is not 100% exact. Okay. So final practical validation is necessary. Either you start with the from in vitro testing, like various cell line studies. You can go with the MTTSS, cytotoxicity study, SRBSS. Either you can check whether your compound, although it is failing the Ripilsky rule, it might be having no toxicity also. So this is just an filter. It is not an exact idea or exact rule that it give you an exact result. Practical aspect is necessary always. So first and foremost, you have to go for in vitro testing. Then you can go for in vivo testing. That will decide a final conclusion. Sir, like you said, log P is very important. What if a natural compound is violating only log P among the other Lipinski rule? We still reject the compound? No, we should not reject the compound only on the basis of log P. As I said you earlier, that more than one or two rule, if violating, okay, then and then you can reject the compound. Okay. You need to check the this database for a natural product. This is one of the nice and curate database for the natural product. Say for example, if I'm checking with the phytochemical here, you just name the compound. Say for example, I'm writing routine. Just click on the name of the structure. You will all get the all parameter or all property again here. All the classification structure you can download from here, 2D, 3D, PDBQT structure, physicochemical properties like log P value, everything, drug likeliness, see, Lipinski rule, violence, Gauss rule, Weber rule, Hagen rule, ADMET properties.
So there is one more tool also, TCMSP database.
Yes, very good database. TC MSP database.
MSP.
Yeah. This is a Chinese database. Where you can get the oral bioavailability also. Here you have to check the chemical name. There are different option has been given. Herb name, chemical name, canonical smileys, cast IDs, target name, disease name. Accordingly, you can check. So here I am choosing the chemical name and I am writing the chemical structure name is routine and searching it, and I'll get the various options. Among them, I'm checking this one, routine. So here you can get the molecular weight, log p value, hydrogen donor, acceptor, and one of the parameter that is oral bioavailability. And this one is a permeability. Kakoto cell line is an intestinal cell line where mainly used to check the permeability. So this is a in vitro tested data. So here you get the permeability of a compound. Percentage of oral bioavailability. So here oral bioavailability is less of a routine particularly. Then BBB, then total topological polar surface area. Okay, so this is one of the also a nice tool that you can use for filtering in case with the oral bioavailability and permeability for natural product only. This is only for the natural product. I'm not talking about the synthetic and other molecule. This database is only for the natural product. Sir, there is no hard and fast rule to consider the number of parameter. It depends on the objective of our study. We need to consider justify. Yes, according to your study aim and objective, you can adjust the or you can use this parameter wisely. To stop a drug from crossing the BBB, we need to make it hydrophilic. But if a drug is not to get absorbed, it should be hydrophobic. How can we balance such situation? Yes, very good question. So to stop the BBB, we need to modify the structure to make them hydrophilic by increasing the polar group. But once the drug is get absorbed, it should be a hydrophobic. So either we need to modify structure in a such a way or in a balanced way so that it can be does not cross the BBB. Or we can modify the final formulation. I can say that final formulation of your drug in such a way that it cannot cross the BBB. Either you can use a certain formulation like liposome. Yes, rightly said, madam said, liposome, phytosomes, neosomes that can be prepared to balance this or control this situation. I hope I answer all the queries. One more question by Vibhuti. Up to which predicted toxicity class is compound acceptable? In a protox, are you talking about protox?
5 and 6.
Yes. If your class or your compound lie within 5 and 6, then it having a no toxicity. Or can be, you have to check particular which kind of toxicity is majorly concerned here. What kind of toxicity is majorly concerned for your compound? Okay, based on that, you can go with the up to 4 also. That is again not a hard and fast rule. So I hope you all are quietly understand pharmacokinetic and ADMET properties and physicochemical property of a drug and how to predict and where you can get the data. Yeah, you can use many numbers of data and take then filter it out accordingly, or you can cross check whether the data from different sources you are finding the same data or a different data. That comparison is also necessary, because these all are the calculation based or algorithm based. So you need to check in a different platform. It is very good practice to check the or confirm the data. Like I give you example, if you check the pKa value of any drug in a different database, you will find a different results. While pK is the most important parameter for designing any formulation of any drug, which is the dissociation constant of any drug. Okay. So during our project with the Jay, we are much or we face many problem to deciding the pK value from different databases. It confuses a lot. So finally, on the literature base, we find that Chemexon is a good software that is gives you a accurate pKa value of a drug or a compound. And we're lucky enough that before the submitting the project or first RSC, the Chemexon was free. And after the submitting the RSC first of his PhD progress report, the Chemexon now become paid. So luckily we collect the data from Chemexon. So this how this kind of situation can be arised. Okay, I think I should have to hand over to session to the Jai, and then we practically help you out after this session if there are any troubleshooting or any difficulty you are finding installation in software. So
Okay.
over to you, Jai.
Thank you, sir. So moving towards molecular dynamics. So I'm sharing my screen. My screen is visible?
Yes, Jay.
Okay. So let's understand molecular dynamic simulation. So what all of you are observing here, that this molecule is moving or vibrating in its frequency. So that's we need to understand. That's what is done in molecular simulation in theory portion. So I am covering the theory portion, theoretical part, that how these molecules bind to the pocket and remains into the pocket throughout the media, throughout its simulation period. So we need here to understand the biology, physics, chemistry, all the concepts related to the molecular simulation. So first we need to understand what is molecular docking, difference between molecular docking and molecular simulation. They are basically not a different thing, they are interrelated things. So molecular docking predicts the orientation or binding efficacy of a ligand molecule in a with a protein in a specific pocket. So as Ghanshyam sir has previously told you, that certain amino acids bind to the protein molecules to show its effect. That can be determined with the help of the molecular docking. While in case of molecular simulation, MD simulation, it is real time or time dependent atomic level view, that how the molecule interacts with the receptor. Okay, so the main point I want to focus on, that molecular docking is the semi-flexible or rigid type of docking. And in case of molecular simulation, it is a flexible and environment based simulation technique. And uh, moving towards the molecular simulation part, I want to tell you that uh, the configuration of a PC or the system is required for molecular dynamics, while in case of molecular docking it is not required higher configuration. And how we can relate the molecular docking and with the molecular simulation is that first we need to initially do the molecular docking to screen the compound or to find out that which type of interactions or which amino acids are interacting with the receptor. I am taking one example that one gene AKT1 is the gene which is most probably seen in the case of wound healing and all that cases. So out of that, in interaction you must see the interaction with the certain amino acids which should be responsible for the activation of the wound healing activity, like threonine and all that type of amino acid should be reflected in your docking poses. Okay. And thus after the docking, the MD will refine and validate that your compound remains into the pocket or may leave the pocket or not, or for how much period of time the compound is remain into the pocket. So moving forward, so what is the need? The one of the most question arises in our mind, that what is the need for the simulation. So simulation or modernization moving or drives the science towards easier use of the molecules. Okay, so computers are not the replacement of the laboratories, experiments, or any other things. They making the things smarter and more efficient. Okay, some of the challenges are there, like time dependent biomolecular behavior, like folding stability measurement and all that cannot be done practically, uh, and it is time consuming and cost effective. So simulation has been made it easy and makes bridges the gap between the researchers wet lab work and computational work. So simulation, how it bridges, the most recent example is COVID-19. So in COVID-19 molecule like spike protein and all, so we are selecting the drug molecule or repurposing the drug or finding the alternative herbal perspective to find out the cure for the COVID-19 during drug discovery or the vaccine design of the COVID-19. So uh, at that time, the docking or the simulation helped us a lot, or the researchers a lot, and reduce the cost and wet lab trials. So what simulation does is reduces the cost and wet lab trials. And in the image, following image, you can see the drug discovery pipeline. So in the drug discovery pipeline, if we integrate the molecular bioinformatics like network pharmacology, molecular docking, molecular simulation, uh, speed up the process of the drug discovery, and thus we can increase the chances of the drug molecule or the new molecule to be selected in the clinical trial, and thus passes the clinical trial and approve for the human use. So some history regarding the molecular simulation. So Karpus was the first person who has done the molecular simulation of the protein. So the molecular simulation is based on Newton's law of physics, that is F is equal to ma, force is equal to mass into acceleration, and Boltzmann constant and Schrodinger equation. These are the main driving equations which are physics equations, or we can say physics laws, which guide the simulation process. So uh, some of the name you are familiar with, like Karpus, Coleman, and Coulomb charges. They have given the force field, MD, charge, molecular dynamics simulation, and quantum mechanics concepts. These are the main building blocks of the simulation that we are currently using for the drug discovery. Moving forward, they have got the Nobel Prize. These three, Martin, Marshall, and Array, are the scientists who got the Nobel Prize in chemistry in 2013 uh, for the development of multi-scale model for complex chemistry system. So, as you can see in the uh, image here, they have done a simulation concept. They have introduced a simulation concept in 2013, for which they have got the Nobel Prize, which integrate the Newton's law and other three laws with the simulation of quantum mechanics. So they have quantum physics, which includes the quantum level of atomic interactions or atomic things which are occurring during the interacting of a compound with the receptor. So in quantum physics and classical physics is the protein. So classical physics means the protein molecules which are uh, there in the system, and dielectric medium is the medium which is the our receptor is there in the human body. So like water or dielectric constant or ions or physical chemical environment, PME we can say, all that things are computationally derived by these three scientists, and they got the Nobel Prize. After that, we have heard in many of the lectures of proteins that amino acids are the building block of the proteins. So with the help of amino acids, these protein molecules are produced or synthesized. So on left hand side, we can understand that what is the main chain or basic moiety of the any amino acid molecule. That is amino group, carboxylic group with C alpha chain with hydrogen attached with hydrogen, and R chain. So changes in the R chain gives us the 20 amino acids. Actually, 20 actually 21. Some of the new amino acids are also discovered recently. So these are the 20 amino acids. So they are interlinked with each other. So how they are interlinked with each other? So they are interlinked with covalent and non-covalent interactions. So aliphatic interactions, aromatic interaction, hydrophobic, hydrolytic, then polar, non-polar charges, they form the interaction with them and form the helix. And based on the alpha, beta helix, beta sheets, and alpha sheets, they form a complex protein molecule, protein structure, which we can see with the help of X-ray. Some of the students have asked yesterday that why we are using X-ray for the screening of the proteins in PDB only. So X-ray is the one of the only method which is measuring the molecule at the atomic Armstrong level. So at the Armstrong level, we can determine the uh, structure parameters of any molecule, and based on that, we can determine the protein molecule structure. So moving towards what is basically a molecular simulation. So in previous slide, you have seen this is the molecular simulation. Okay. So this is totally based on physics and other parameters. So here I am going to brief you that what is basically happening in background, what these softwares are doing in background. So molecular simulation is based on the three equations that I have shown you previously. Newton's equation is the most basic equation. F is equal to ma, force is equal to mass into acceleration. So in molecular simulation, including water molecules and all the molecules in the protein molecules and even in our ligand molecule. For each time frame delta t, and for each atom for the time interval of initial time and the final time t, I and t, it determines its position, velocity, acceleration, force, and potential energy with the help of these equations. And with the help of energy function, we can determine the various force field. So you have heard that we are using different type of force field for the measurement of different used by the different softwares. So here is the main equation which determines the different force fields. So here force field. So here the V is equals to, V means energy. So here the first equation over here represent the bond stretching. Then second here plus addition with the bond angle. This is the angle equation. And this is the type of interaction, that is hydrogen bond plus van der Waals equation. And this is the charge equation. Okay, so based on these constants like K, V, R, and charge constant is constant. So based on changes the constant, the type of force field may change. So I will show you which type of force fields are available. So changing the force field changes the equation, and just thus changing in the equation changes the atomic position, velocity, acceleration, force, potential energy at real time. So the selection of the force field is one of the most important thing in case of molecular dynamic simulation. So I have explained you. Many of you are aware about this wagging, twisting, and stretching, bending, all these properties of the atomic molecules, the movements of the atoms. So this is again the same thing. So same concept, but here I have told you that it will generate this type of file and calculate for each molecule, and after that we will get a trajectory file, which is trajectory dynamics folder or file, which will be generated with the help of the softwares, different softwares. So based on the force field, so we can get the trajectory file which will give you the simulation results. Okay, after moving forward, that simply what is if you want to understand what is a force field, force field is the intermolecular and intramolecular interaction between the molecules. So it is the energy equation. Okay, so this is the basic understanding that how force field or energy in the terms of molecular simulation can be understood. So as I have told you previously, that force field is a energy function, and it will determine or calculate intermolecular or intramolecular, or we can say interaction between the atoms. Okay, bonded, and it may be bonded or non-bonded. Thus moving forward to the equation, energy is equal to energy for stretching plus bending plus torsion plus electrostatic interaction, uh, E van der Waals forces, and hydrogen bonding. I have explained each and every some of the parameters in the which type of energy can be calculated in case of stretching, bending, and all. So one of the main parameter which we need to understand is the van der Waals forces and hydrogen bonding. So repulsive and attractive forces can be calculated with the help of E van der Waals, that is E van der Waals forces, and in hydrogen bonding we determine the hydrogen and hydrogen bond donor and hydrogen bond acceptor of a amino acid and a compound interaction with the help of molecular dynamic simulation. Okay, this all comprises of the physics and all the parameters of the chemistry, quantum mechanics, and all. So initial in molecular simulation, the force field is calculates that here I have explained you that how this simulation works. That initially at the atomic level, it will calculate the molecular forces acting between the atoms and move each atom according to their forces, and advanced simulation time with 1 to 2 femtoseconds. So we have heard about seconds, then microseconds, nanoseconds, picoseconds, and then femtoseconds. So molecular simulation, molecular dynamic simulation will calculate the uh, changes, which is position, velocity, acceleration, force, potential energy of each molecule at the time frame at the 1 to 2 femtosecond changes. And thus at the 1 to 2 femtosecond changes, it will calculate the molecular interaction and all that. So collectively, these parameters are called force field because they are described in contribution of the various atomic forces that govern molecular dynamics. So this is the main heart for the molecular simulation theoretical point of view. And moving forward, so in case of molecular dynamics, we constrain the molecule into a periodic boundary. So what is periodic boundary? So here I can show you in the image. This is the periodic boundary. You will observe only this white part. Okay. And other are periodic boundary set by the parameters which we have given to it. So what happens here, I want to understand you that sorry, you have seen here that the molecule is vibrating at its own frequency. So or the drug molecule is into the pocket. Here you can see the drug molecule is remain into the pocket. So what is happening here that I want to tell you, that only we can see this window. We are not able to see these windows, or as we are simulating it in a 3D plane. So what happens here, if the molecule stretch and bends and goes towards the this chamber or this direction, so it will, the simulation boundaries constraint will show it to be vibrated in the mirror direction. So if the molecule goes in right hand corner side, it will show you the molecule will come from the mirror image that is left and bottom side. So this is the main reason why we do the periodic boundary, and this is the first and foremost step that we constrain the periodic boundaries for the molecular simulation. And if we do not do this, then we cannot visualize that this compound is how this compound is or docking or the complex protein ligand complex is simulated. So what do we get from the MD? You have seen this type of graphs. Okay, so each and every graph tells us the different story. So here the RMSD, root mean square deviation, RMSF, and surface SASA, accessibility, radius of gyration. These are the main uh, key results which we get from the MD simulations. So these parameters predicts that our molecule or drug molecule remains into the pocket for the simulated time period, that is 100 nanosecond. So different researchers uses different type of simulation timing. Okay, so we can use it for 100 nanosecond, 300 nanosecond, or 1 microsecond as per your system configuration. Okay, so moving forward, I have told you that different types of force field are there. So why MD simulation? Why do we need MD simulation? To capture the real-time position of the atomic and confirmation that docked molecule is the remains into the definite site or pocket or not. Okay, so molecular simulation can be used to confirm that your drug molecule is there, is remaining to be there or not. If the molecule is removed from the pocket, then the molecule cannot be stable in real life or on in the human body. As in human body, the molecules are vibrating at its own frequency, as I have shown you in the video. So the it mimics that environment or that kind of conditions into the simulation conditions, and thus confirm or validate that your compound is can be used for further clinical or in vitro in vivo studies or not, or clinical trials or not. So this is the one of the first step to for the drug discovery before doing the clinical or preclinical studies. Okay, so these are some of the force field, and these are the packages. Charm force field includes charm and ember. Then ember force field includes ember force field and gromax. Gromax uses ember, gromax or opals, opls, and charm force field. And NAMDA using charm, ember, and gromax. And desbond uses oplas AA. So these are the common force field, and so some of the softwares are paid. So we are using the free software, which are one of the free software is Gromax, which uses the this force field, and most commonly used force field are charm. So the question arises in your mind, how we can select the force field. So based on your protein molecule, you should select a which type of force field you should do, or target environment. So for example, if you are targeting cancer, for example TP53 gene, for TP53 gene, the force field commonly used is charm force field, charm 27 or charm 36 force field, based on latest edition. So this force field which is used can be can mimic the environment which is there in the human body. So based on that, and based on the software's configuration, we select the force field. So moving forward, you have seen this Ramachandran plot everywhere, that what is the protein, and what this is the basic blueprint that how this protein molecule should see. As in yesterday's lecture, Sir Ghanshyam sir has showed that the Ramachandran outlier should be there towards zero. Okay, it so here I explain you that what is Ramachandran outliers. So here is the graph, phi and psi molecule, psi angles. So phi and psi angles are the angles of beta helix. You will see any any protein molecule in this form, that this helix type of structure is alpha helix, and its plated are beta sheets, so or beta helix. So these are the conserved region. Given this graph is given by Ramachandran, one of the greatest scientists in the history. So beta sheets, beta region, then right hand alpha helix and left hand alpha helix region. So what are the sir has told you, it should be near to zero. Yesterday sir has told the Ramachandran outlier should be towards zero. So what does he mean? That Ramachandran plot, most of the amino acids or the outliers should lie in these regions 1, 2, and 3, which is represented over here in red color. And if the Ramachandran outlier is towards 1, sir has told avoid to select that type of protein molecule, because outlier is outer the Ramachandran outliers, so in yellow region or white region. So we should not select that protein molecule for docking, and as well as not for molecular simulation, at its as its stability is not there in throughout the medium. Okay, so moving forward, this is the molecular dynamics workflow. There is system setup. I will explain these steps by performing hands-on. So I am briefly explaining the what is the meaning and what is the theoretical part, that how we can do we are doing in simulation part. So system setup. So we prepare a 3D molecule as we are going to perform the molecular simulation in a 3D chain. Okay, 3D XYZ axis. So XYZ coordinates of the protein molecule and ligand molecule is to be taken, or 3D coordinates will be taken, and step 2, solvation. So here the force field plays an important role. So force field, you can see the box. Any doubts? Hello, hello.
Please continue, Jay.
Okay. So in solvation, you can see here orange, violet, and blue color of balls, or we can say molecules in the this periodic boundary. So what does this represent? So actually, blue molecules are water molecules. These orange molecules are the calcium ions, and violet molecules are the sodium ions. So in our human body, these are the same environment which is mimicked in this molecular simulation. Okay, and then energy minimization. So we compact this molecule into the with least energy. So we have studied in quantum chemistry and quantum mechanics that the molecules are most stable in its minimized state or stable state. So molecule are first given to a minimized state, or we can say most stable state. After that, we equilibrate using the simulation work with the help of NVT and NPT. So what is NVT?
I think he might disconnected. Just wait for a minute.
Hello, sorry for the disconnection. My screen is visible.
Yes, Jay.
Sorry, sir, for the inconvenience.
No,
So
continue.
I think I have left over the step 4. So I have told you that at the energy minimization is the most stable state at which the molecule is most stable. So after the molecule setting into its most stable state, we excite it, or we mimic the environment into the as the human body. So here are the two things, NVT and NPT. What is NVT? Is the temperature. We give the temperature, exciting temperature, and the normal human body temperature, which is to be given into the simulation environment, and for the specific complex or a protein molecule. And in case of NPT, we will provide the particular pressure for the protein molecule to be simulated, and after that, from step 1 to step 4, we will give all the uh, constraint to the protein molecule or the complex molecule that you have to simulate in this environment only. After that, we will perform the production MD or MD run, final MD run, which is from time t0 to time t. So here time t means 100 nanosecond, from 0 to 100 nanosecond and all. Okay. After the MD simulation is done, the MD simulation results trajectory analysis includes, I have explained you before, and we will also perform in the hands-on training also, that RMSD, RMSF, radius of guidation, number of hydrogen bond, SASA, solvent accessibility surface area, and MMGBSA, MMPBSA if possible, and all. And post simulation processing is the processing of the trajectory files for certain molecules and visualization and interpretation of the stability of the molecules. Okay, so here the MD run theoretical part is done, and at the practical session I will continue with the practical hands-on training. If you have any questions, please feel free to ask the questions. Thank you. Hello. Yes, Vibhuti has asked a question. Sir, we can able to run simulation on our normal laptop. It totally depends upon your system configuration. If you are using a Windows system, Windows operating system, and it is having certain 16 GB RAM and graphics card, so you can easily run the MD, but it may take a longer period of time. So it may take a month, and for a month you should keep your computer or system on. So you can use a simulation working station to do the simulation, a molecular dynamic simulation.
Mostly the molecular simulation is done on a Linux system. Okay.
Yeah.
Mostly all the molecular dynamics has been done on a Linux platform, not on a Windows platform. Okay. So ideally we should have to run on a Linux platform, because it is a command based software.
Actually, my next session is time is not permitting me, but you need to install the Linux system in your Windows if you are Windows user, and for the Mac users, you do not need to install the Linux system. So I am sharing in the chat box the link, or we can say Ubuntu. Ubuntu to be downloaded from the Microsoft Store. Okay, and after the downloading through Microsoft Store, you can use the following video. I am sharing the link of the video, YouTube video, by which you can download the tutorial, which is used to download the GrowMax software into your Ubuntu system or your in your computer. Please make sure that your system or the computer or laptop, whichever you are using, are compatible to handle this simulation. If not, it may hang, or it may damage your motherboard and whole PC also. Ghanshyam sir. Yes, sir.
So my advice is to up to the protein preparation and fourth step up to the equilibrium, you can run in your Windows system. Okay, how to do run that? I'm just giving you an overview or just an example. I'm sharing my screen. I hope my screen is visible to you. As Jai said, you have to install the Ubuntu through the Microsoft Store. Okay, so here is the icon of Microsoft Store. Go to that store. My screen is visible,
Yes, sir.
and
Yes, sir.
right here Ubuntu. You can select Ubuntu Pro or Ubuntu for Windows here, or Ubuntu for VSL. VSL means Window System. Then you have to just, I'm already installed it, so I will not get the option here. So you will get the option like get it. Just click on the get it, it will automatically install in your system. Okay. Once it is installed, it will appear by this icon. Okay. By clicking on this icon, your Ubuntu will work in a Windows system. Your Linux will run into the your Windows system. Through this,
Here, sorry, sir.
yeah.
Here, I want to tell you something. I think sir is having the latest version of Ubuntu. So, sir, please share that Ubuntu terminal once again. Okay, it is showing hashtag. Okay, if hashtag is there, then it is used for Mac also. Okay, integrated with Mac. And if the hashtag is not there, or the forward arrow is there, it can be used for the Windows system also, or Linux system. So some of the codes provided you the I have provided you the link. So in some of the codes may not work run here. So as per the uh, system, you should have to change some of the codes to run the download Gromax. Okay, yes, sir.
Uh, yes, Anuchal, you can use a dual boot system for your system. You can use both the OS, Linux as well as Windows. So at the time of booting, you can select the whichever the OS you are requiring. Okay. But the prime most requirement for the molecular simulation is having the high RAM, having a good processor. At least right now, if you are going for 100 nanosecond, at least i9 processor is necessary. i9, i5 is also sufficient. i7 is also sufficient. But latest one is i9. It can be work fine. The core of the or thread of the your processor must be higher. RAM is also required. Okay, RAM must be at least a minimum of a 64 GB or 32 GB. Higher is good. Okay, so and GPU, graphic processing unit, GPU is required for simulation. As Jai explained that uh, the entire molecule simulation is tracking the trajectory of each and every element. Okay, so to tracking of each and every element, say for example, if I am having the protein which having the 100 amino acid, and each amino acid having the 20 elements, so 100 multiplied by 20, so mostly 2000 elements should be tracked at a time in a femtosecond level. Okay. So for that tracking and reporting the trajectory of each and every element at quantum level, at femtosecond is actually quantum level, it require a GPU. CPU alone cannot do or tracking these entire elements. They're tracking the velocity, force, energy, wagging, twisting, everything is tracking through this GPU. So GPU is required compulsorily for the molecule simulation. Higher the GPU configuration, lesser your time to be completed the molecule simulation. So better to you have to have a workstation, not a simple system. Workstation is necessary. Should we have to download the latest Ubuntu package or older one? It can be work both. Latest also can work and older one can also be work.
Also depends upon your Python environment
Yes.
or
Here
which
you
type
can
of Python you have.
here you can write the command sudo update.
Sudo apt update.
Yeah, sudo apt update, and you can update your system any time. I'm not updating it because my system is configured as per the requirement. So I'm not required to be updated. In case of modular software, when I want to install some license key is required, what to do? You have to register first on a modular for yourself. Once you registered, you will get license key in your email address. That license key will work for every update and every version of software. I might not be familiar with right now which modular version is working, but in my system, I have installed modular 19. So for that, you will receive the key in your email address once you registered. So once you have installed the Ubuntu or Linux system, then you have to install the Gromex software, because Gromex is compulsory to prepare environment and to run entire simulation and all the force field which are included in this Gromex software. Why we using Gromex? Gromex is a open source software, free software, and highly accepted in publications, and which unable to use a various force field like charm, amber. Okay, these are well reputed force field which are mentioned in almost all the publications. That's why it is validated force field, and that is included in a Gromex. That's why Gromex is a most suited open source software or open source package for your simulation. For that, you need to check the video which is provided by the Jai. By that, you can easily install the Gromex in this Linux system.
In case the latest Gromex cannot be configured with your Python system, you can use the older version also. Do not use the most older version like 2019 or all. At least you should go
23,
to 23. Yeah,
2023,
2023 is the last version which you can use, as some of the force field supports the last version which is 2023. So if 25 is not working, you can go till 2023 or 2023. Do not install other older versions, please.
Yeah, Vibhuti has asked again question that regarding drug discovery studio, that your older version you required. Okay.
I will share the file, downloading exe file. You can directly install it from the exe file or executable file in your Windows system.
One question is there.
Uh,
Okay. Will molecular
yes,
dynamic
sir.
simulation run efficiently on a laptop with the AMD Ryzen series processor and NVIDIA GeForce RTX GPU? Yes, it can be run. Uh,
What is your RAM?
RAM capacity or GPU capacity you have already, like GPU, like GeForce RTX must be of a uh, at least uh, 8 to 16 GB that can run
Okay.
easily. 4 GB is
Yes.
can be run, but it will take the time. Your time will be more.
Yeah. Okay. I want to tell you something that if you run MD simulation in your system, then at the time of NVT or NPT analysis, you will see your system performance. If it is sufficient enough to be done or keep your system on, then you can run your MD simulation in your system or the workstation.
At the time of hands-on, we will explain you each and everything, and you can decide yourself whether this system or this simulation will run in your laptop or not. You can easily decide. My advice is do not start immediately to running MD on your system. First, understand everything, each and every aspect, okay, limitations of your software, your system. Otherwise, it will immediately crash your Windows. We do simulation in our, we have a workstation. Okay. We can go up to 100 to 200 nanosecond in our workstation, which cost around the lakhs of rupees. Okay. We have our system in our institute. Okay. So we will give you a demo of that particular environment into this workshop. Jai, you can answer the last question easily. Abhishek.
And it's, yes, sir.
Any software which can calculate drug release from resolution study and also Cmax, Tmax, AUC, MDT, PKPD modeling? Yes. You can
Yes,
use
we
a
work
add-on.
DD solver.
Uh,
Oh, sorry, sir. Sorry, sir. You continue.
yeah, you can use a DD solver add-ons into the Excel. Okay, by
Or PK
using that.
solver.
Yeah, PK solver that is easily available on online, freely available. You can add on, or it is actually a macro. You can add into your Excel, and you can calculate all these parameters. Shivraj has
Uh,
asked, can we do it online software directly? Yes, there are a certain server is available to running the MD simulation online. Okay, you have to submit your job and you have to wait. You just have to give a your complex of your ligand and protein after docking. Rest of the thing will be done by the server. Okay, but the limitation of that server is that this server will give you a only a sometime 10 nanosecond or 15 nanosecond simulation only. Other extra time you require, like most of the publication ask you 100 nanosecond simulation. Okay, for that you have to purchase the credit, and that cost you a lot. I think I'm giving you a right answer, Sivraj. There are certain various GPU available online. You can purchase it like Google Colab. Google Colab is also one of the tool you can use for the molecular simulation and dynamics. Okay, you have to run set up a Python setup on a Google Colab. You might be just let me show you what is Google Colab. Google Colab is mainly work on the principle or on the platform IDE called as a Jupyter. The Jupyter notebook is mainly used for as a writing a Python environment. So this is a Google Colab. You can start coding or start a writing a code from directly from here. Okay, it is directly associated with your Gmail account. Okay, but before that you need to connect with the system or connect with the GPU or TBU, which is provided by the Google uh, platform or Google server. So here you have to change the run types. Okay, which environment you required, Python, R, anything you are required. Mostly the Gromex system work on based of Python. Okay, so here you have to use the GPU for your system, like this T4 GPU, then CPU and V5 TPU. These are the free one. Okay, you can use it for a free, but for a higher GPU, you need to purchase a credit. You have to pay for your using of this GPU, which will provide by the Google. Okay, one of the drawback is that at every 24 hours, this system will refresh everything. So it will lose your simulation at after 24 hours. So this is the one of the drawback of a Google Colab. Okay, you can purchase more credit and you can use it wisely. Nowadays, NVIDIA is also providing the GPU. NVIDIA platform are also available for the GPU rental, but it costs you a lot. Okay, NVIDIA providing you a GPU sources. One of the, it's a docking platform is also available, which is called as a diff dock. You can use a diff dock model and you can run a docking on this platform itself freely. But you can run only docking. For GPU, you need to pay. Means you get the rental GPU here. But I advise you that if you can, your university or institute may provide you a good system, then you can run it easily. Otherwise, you can collaborate with the institute where the facility is available. Like we are having the good workstation. You can collaborate with us and we can help you out in running simulations. One more question that I have performed only normal mode analysis through iMode software. Is it okay for publication? That is, I'm not actually familiar with the iMode. If you can share the data to me, then I can explain. I can suggest you whether it is sufficient for publication or not. If any question furthermore, or if you're having any trouble for installation or anything, please let us know so we can solve it right now. Oh, previous software we have provided explain you in earlier. Okay. What is your question, Priya? Sorry, the call was ended when the answer my question. Kindly repeat me. When will you teach us about the network pharmacology software in detail and how it used to be? So as per the schedule, Dr. Sumit, I think tomorrow will be the session for the network pharmacology.
Yes, sir.
Yeah. So Priya, tomorrow we will explain you how to handle this software and how to interpret the results. So Dr. Sumit, shall we conclude the session or
Yes, sir.
question?
So I thank you very much to all the participants as well as our speaker Dr. Gancham sir and Mr. J for such an informative and engaging session. Your explanation of pharmacokinetic and molecular simulation has helped all of us to understand these important topics in a simple and practical way. We sincerely thank you for your valuable time, guidance, and continuous support. I would also like to thank all our participants for joining today's session and for your active participation. On behalf of Thyme Phytobiomed Private Limited and Adgrow Foundation, thank you once again for being with us today. We look forward to seeing you all tomorrow at 7 p.m. Until then, keep learning and keep exploring. Thank you, everyone. Have a wonderful evening. Good night and Jai Hind.
Thank you, everyone.
Thank you, everyone.